Yes, sir, you're live.
All right, very good morning, everyone. All the students of MOOC course on Interactomics. Today is the third and the last live session we have. I think soon you're going to finish your course as well. So I hope you're finding many technology which you are learning in this course to be quite useful and interesting. And the live sessions, we are trying to give you some thematic way of understanding so that you are able to. Very good morning, everyone. Able to get the concepts better, and also you can get a feel of how the things can be done in the lab, right? So after two thematic sessions, today we are going to focus on more of the technologies which you have learned in more detail about protein microarrays and some of the other label-free biosensors, how they can be utilized for various biological applications, but we will focus some of the clinical applications. Along with me, there are various TAs who are going to show you the things from our Proteomics Lab IIT Bombay live. I'm currently at University of California, San Francisco, but still, happy to connect with all of you live for this session. So, since we have been taking examples of COVID-19, just to remind you that during the COVID-19 time, a lot of knowledge was gained uh, just by unbiased approaches, right? We had no idea at that time that how this virus works, and even still, we are again seeing the surge of COVID-19 in India right now. So, to really understand this virus or any kind of pathogen or infectious disease or even long-term impact of it, we need more of the holistic approaches, right? And that's where some of the multi-omics technology have been quite helpful. Of course, it started with NGS and developing the primers and RT-PCR kits, understanding the structure with electron microscopy, looking at different type of peptide protein with mass spectrometers, and then trying to look at more of the candidates for the vaccine development and the what makes them more relevant for the the humoral immunity and those stuff. So again, then protein arrays and various type of high throughput screening approaches have been quite helpful. Different type of data analytics has been quite important. So again, this is just one of the examples that how various technology which you study in the course could be utilized to understand the unknown entities like this. So let me just refresh you about protein microarrays. Of course, you have studied in the course. I don't want to give you again a big lecture on this, but remind you that talk about protein arrays. Unlike the oligo arrays, we are trying to look at proteins in different ways, whether antibodies to be printed or the purified protein to be printed, synthetic peptide to be printed, or even one could take the cDNA clones or the any type of DNA material or PCR based product and try to make the protein on the chip itself with the process known as cell-free expression-based arrays. And you have studied in detail about NAPA arrays and various type of arrays, right? So again, this just summarizes that different approaches can be taken forward. Uh, so when we think about applications of protein microarrays, uh, just we are trying to give you a live application because now you can see there are different ways of utilizing these chips. One could print all the human proteins on the array and look at human disease or look at the drug interaction or the protein-protein interaction, right? So a typical kind of protein microarray is involved that all the proteins of a given organism or any entity has been printed on the chip in a very high density format. But think about DNA versus protein. It is not very straightforward. We have to really uh, think about all the challenges of keeping the protein intact and the printing of the protein properly. So there are many challenges, but still protein microarrays are made for different organisms in human. And one which we use in the lab is the human proteome array chip, HuProt chip, which is quite good representative of all the human proteins. So on these chips, one could do different applications as you have been studying in the course about protein-protein interactions or looking at different small molecule and protein interaction, their binding, inhibition, or looking at the modification of the how different type of PTMs could be studied, enzyme substrate analysis could be performed. many inhibitor screening could be done. So many of these things can be done on these kind of protein arrays. But what you may not have thought about, like one could also study the virus like SARS-CoV-2 on the protein arrays, and especially what we chose here in this case of the peptide array. So we collaborated with this company from Germany, Pepperprint Array, and they have made these tiled arrays where all the proteins of the SARS-CoV-2 virus, different peptides of those, let's say 15 amino acid long, these peptides have been synthesized, and they have been, overlapping way, they have been printed on the small glass slides or the chips. They can represent the entire proteome of the virus. So now this particular proteome which we are talking is essentially represents the entire peptide compositions of all the proteins of SARS-CoV-2 virus. So with this, now I'm going to show you how this type of experiments could be performed on protein array platforms, right? So let me just give you, this video, which is essentially we have shown from the lab how you can do the peptide arrays to screen for the SARS-CoV-2 virus.
Today, we will be demonstrating peptide microarray technology. First step involves reagent preparation, namely washing buffer, staining buffer, and Rockland blocking buffer. After preparing the reagent, we move on to patient tray assembly. Unscrew the incubation tray by slowly turning the screws diagonally. We will be using two blank glass slides as balancers, since today we will be using only one microarray chip. This is a pepprint microarray chip. Slowly take out the chip using plastic forceps and place it on the incubation tray. Align the chips properly onto the tray. Now, allow the gasket to be placed over the slides. Remember, the glossy side of the gasket on the downward side. Align the silicone gasket over the slide and tightly seal the incubation chamber using the screw. Remember, tighten the screws only diagonally. This will ensure add 2 ml of staining buffer onto the chips. This step is called equilibration of peptide microarray chip. Please note that we are not touching the microarray chip at any point. Now allow the equilibration process to happen for 15 minutes. Now, activate and remove the buffer and add 2 ml of blocking buffer. Slowly add the buffer onto the slides. Allow for 30 minutes of incubation. The next step is sample incubation. To the staining buffer, we will add patient plasma at a dilution of 1:200. This is the patient plasma. Now slowly overlay the diluted plasma over the slide and incubate overnight. This will be done in a cold room at 4 degrees Celsius at 140 RPM on an orbital shaker. On the next day, remove the incubation tray from 4 degrees Celsius and aspirate the solution. Now add washing buffer thrice onto the chip. This step is done 3 times in the dark. Again, completely aspirate the washing buffer and add secondary antibody. Layer of dilution of secondary antibody 1:2000, and then slowly overlay the secondary antibody on the microarray chip. For 45 minutes, allow the incubation to happen at 140 RPM at room temperature. Make sure that you do not expose the slides to light at any given moment. Now remove the buffer and wash the slide thrice using washing buffer. Now gently take out the slide and put it in dipping buffer. This is done to wash away all the salts that may be over the peptide chip. Use a pressurized stream of nitrogen. Finally, we will scan the slide. This is the microarray scanner, GenePix 4000B. We are using manufacturer recommended settings for scanning the slide.
All right, so I hope you got an idea about how to perform the microarray experiments. But the next major challenging aspect is the data analysis, right? Like how you can take that data and try to now make sense of that information, because we have really high throughput data set which can obtain, and a lot of features which are printed on these arrays include different type of control spots, control features, and the actual protein and peptides. So you are going to ensure that the non-specific background and the noise has to be removed and the real signal has to be extracted out. But all that looks very simple, but actually requires a lot of you know many times good programming skill if you have. Otherwise, even you have to spend a lot of time on the Excel sheets to really expand and do statistics to make sense of the data. So let me now invite Arko Ray. He's a PhD student in the lab and jointly from Electrical Engineering Department and BSB. And he's doing quite much work on the data analysis aspect of the microarray. So I think he is going to now talk to you about data analysis of these peptides and different type of microarrays. Arko, over to you now.
Good morning, everyone. Now I'm going to tell regarding the micro data analysis procedures. So let me share my screen.
Yeah, we can see your
Yeah.
screen now.
So okay. Thank you, sir. Thank you, everyone. So myself, Orko, I'm doing PhD under the supervision of Professor Sanjeeva Srivastava. Now I'm going to show regarding the micro data analysis procedure. So in the micro data analysis, data extraction is really important. This is the basic schematic experimental workflow. I think you have just gone through actually the steps that how the biological assays has been done with the microarray chips. So this part is the GenePix scanner, which you already seen that how the chips after the biological assays, how the chip inserted to get the microarray image. Now I'll actually give you the live demo regarding the data extraction, because that is the very important part, because data extraction, if our data extraction is not proper, then we cannot go further for the data analysis. So now I request Avinash to come on the live. So I'll give you the idea regarding the data analysis, data extraction part. So sir already mentioned regarding the so
You're
this
not
is
audible,
the
Arko. Yeah, okay now.
So this is the microarray chip. So it is the protein microarray, as already sir has given you some idea regarding the protein microarrays as well. So I just need to give you some idea regarding this. This is a normal glass slide. So at the thousands of proteins are printed. Now we need to see the chip into the scanner. After inserting that, how the chip is looks like. So I will give you the two different chips. I will show two different. This is one chip came from Huprot lab. Huprot, this particular CDI lab. This is called human proteome arrays. But as also sir mentioned regarding the peptide microarrays, so there you could see the another chip. The chip is little bit different. It looks like little bit different. So this is kind of a peptide microarray. So there actually these are the microscopic slides. So after inserting that into the particular scanner, so this is our scanner. So this is the tray. So if we just insert this, you can see that already the scanner is on. So now I'm So now I need to show that how this can actually actually the images looks like. So now you could see. So this is particularly the yeah. So this is kind of a particular image, microarray image. So these are the Huprot arrays. These chips for the protein microarrays, we usually observe this microarray chip. So these are the GST, practically the proteins are probed the GSTs. So usually for the microarray chip, usually go for the two scan, the 532 and 635 nanometer for this particular protein microarrays. So for this green channel is compatible for the QC, that is the quality checking, followed by another for the autoantibody profile. We usually go to this another particular scanning, that is for 635. Now you could see so the spots are quite in the particularly the red channel. So in the red channel, you got this kind of image. This is for the 635. So for autoantibody profiling, we usually check this particular microarray chip with the 635 nanometer wavelength scanning. Now, after doing that, as we are concentrating on the peptide microarrays, so now I also want to show, as already sir said, regarding the tile formation of this particular spots. Now we could see if we very closely if we show this. So these are kind of actually this is called the epitopes, and what sir said regarding the control spots like the HA and polio. Now you can see these spots are really prominent over here. So these are called epitopes, and after proper data analysis of this particular peptide microarray, our interest should be to come up with this particular data set, and we want to claim some kind of immunogenic peptide that I will show in detail of my PPT slides. Now, if I want to give some idea regarding this is called the GenePix Pro. So this GenePix Pro is compatible with this particular GenePix scanner. So this is actually you are using from just to scan the image and get the image out of it. But after that, if we want to perform the data extraction, we extracted another software that is called the Mapix. So now this is for our another system. So now you could see one particular grid file is associated over here. So this grid file is nothing but it is we usually call it as gal file. So that is called GenePix array list. So it is nothing but the annotation of the peptides. So when we just map this peptides as CSV file, when we just incorporate that into the particular portal, just a software, you could see that it is completely on the particular images. So every spots point of view, you could see it is gridded with the particular file. So here we want to grid the particular spots very minutely, otherwise we will lose the data out of it, which is not at all acceptable. So after that, what is our next data set is required for data analysis? That is called GenePix result file or GPR file. I just need to show in this way. So now you could see this is called the GPR file. So this GPR file has the lot of ideas regarding the GALs, which I already mentioned, that the protein peptide annotation followed by block, different blocks, where the blocks are there, row, column, everything, because those are important for finding out the particular peptides when you go to the programming part. Now you could see these IDs of the peptides as well. So this, in these IDs you could see the different type of peptides are printed, and this could be taken forward. These are the GPR file could be taken forward for finally into the programming part to get the whatever our interest, like the immunogenic peptides or immunogenic epitope, followed by discriminatory peptide, discriminatory epitope, all these things. Now I want to come up to the my presentation slides. So I think, sir, my screen is visible. So yeah, so I already given you the detail regarding the scanning of the paper print array, so that you have just seen. So for this work, actually, we recently published one paper based on this SARS-CoV-2, the COVID-19 samples we have dealt with. So there, our interest was to find out the immunogenic epitopes for both IgG and IgA. So for that, the scanning has been done for both 532 and 635. So for getting the exact signal out of it. Now, GAL file placement, this GAL file also could be made in-house. So we are experienced to do that, because sometimes we usually this GAL file has been came from the different companies who actually provide this kind of paper print arrays. They sometimes give us this particular GAL file, but we are also in-house in our lab only. We prepared this GAL file. Then what is the GAL files I already told? This is kind of an annotation of the peptides. So now Mapix has the compatibility issue. So I have already shown the Mapix software. So each has the compatibility issue. So when we just incorporate that particular file into the Mapix, you could see this kind of grid, which actually give us our time, which actually save our time to because it is helping us to extract the data using this particular particular GAL file. So after doing that, now it is the time to show you the GPR file, because these are the connected. So first of all, data extraction. In the data extraction, GAL file is important, the InPix array list, followed by GPR file. Why GPR file is required? Because finally we need to go for the understanding of the immunogenic peptides or epitopes, what I told you. So biologically, the data analysis team, how they proceed. So they proceed using the instruction of the particularly like the who are doing the experiment. So they have some kind of thought that what kind of marker they really interest for which kind of markers, because those can be taken for the finally for this kind of making the vaccines from the pathogen from the laboratory. So this would be important markers. So as far as their idea, now data analysis team usually proceed further. But the basic thing is that between the protein and peptide arrays is we want particularly the GPR file, because it is kind of the annotation of the particular proteins or peptides, followed by the intensities. Which intensities we are really looking forward? Mean, median intensities of the foregrounds as well as the backgrounds. So now this file to be incorporated into RStudio. Why R? Because other softwares are also available, or other coding platforms are also available, or Excel sheet also sir mentioned that, because it is time consuming. Now if we can read, just I know, if you see this my note part, read any number of GPR files. So instantly with a TSV file, if we make a TSV file with the particular name, with the particular programming part, it can automatically read all the GPR files. So now after so this is kind of a GPR file. I have shown that for the peptide arrays. Now after reading the GPR file, next very important part is background correction. Why? Because there are several models are there. I wanted to show you that for the standard background correction, then why the Cooperberg, Edwards, Normex plus offset, why these models came up. So for that, standard for particular the model, this is normal procedure. It can be done from the Excel sheet as well. But problem is that, are we really taking forward the weak signal or not? Because sometimes weak signal also could be a very interesting part for our analysis. So for that, let's say I'm giving you a normal mathematical part. Let's say we are subtracting the foreground intensity with the background intensities. So let's say the background is little bit more than the foreground. It will give us the negative intensities. Now, definitely to remove the skewedness of the data set, we usually go for the logarithmic conversion. Automatically, when you go for the logarithmic conversion, it will give you log of any negative value will be showing the undefined. So that is not at all acceptable. So researcher came to actually try to do the try to came up with the different strategy. So itself background correction is the research part. So they came up with the different models. So finally, maximum researcher at this point of time with this data analysis of microarrays for protein and peptide, they prefer the normex plus offset, because normex is a convolution model and offset is that we have the provision to strengthen the signal. So this is the logic of the offset. So we may prefer to give the, for our previous analysis, previous work on the peptide microarray, we have given the offset as the 50 to strengthen or boost the signal. So finally, this kind of libraries are important. And why RStudio? This is the fact, because we need to know regarding the Lima package. So that provision only we have in the R language, R programming part, and we need to go for the background correction. So only for background correction, if somebody is really interested to find out the G score or like that, any coding part is important, but we cannot skip R because of this background correction libraries, which is available in the Lima. So after that, you could see just automatically after doing that, array 1 corrected, array 2 corrected, means on the coding itself, you could see this kind of correction has been done. So after the background correction, what is the next call for the peptide microarray? So we need to go for the different QC strategies like interchip reproducibility, intrachip, and interchip. So why interchip? Interchip, if it fails, it comes to the particular manufacturer point of view, because the concept is like that the proteins are printed or peptides are printed in duplicates. So if the duplicates, one is actually spot is binded with some kind of its probe and analyte. So why not the second one? So this is nothing but the particular intrachip. So spot 1 sorted spot 2 deviation. So it should come close to each other. So definitely intrachip reproducibility should cross the value of 0.9 minimum. So if we could find it approximately more than 0.9, then we go proceed further. Otherwise, we definitely rethink about the chip and we consult with the companies for that. Next, coming to the interchip as well, because it is a day-to-day reproducibility, we need to vary, check. So for that also we go through it. And sometimes also another strategy is required. Sometimes it also not give us much confidence. So in this paper, we have seen the letter value plot. So this is nothing but the distribution of the data, how it is looking like. So if the distribution of the data, let's say this, you could see the P7 sample more or less skewed. So why should it? Because other data are really deviated, but it is a skewed sample. So we should rethink it. Are we going take it to proceed with it, or we should discard it? So we have discarded this sample. So you could see P7 and P40 because of the skewness of the nature. Now, after this particular QC has been done for the peptide microarray, as I'm talking regarding peptide microarray, G-score is the very important scaling strategy or standardization strategy to further the analysis of the data. For that, how the Z-score, so this is, they are also sometimes mathematics is also required. So we need to know the intensity of every spots that we know from the GPR file, mean intensity of the peptides, all the peptides printed, and finally standard deviation. If we could recollect, if we could come up with this all the means particular equation, if we take up into the R language, so it will give us the Z-score of each and every peptides. So after that, if it is done, now why I already told you regarding the immunogenic peptides. So for immunogenic peptide, it's a very important part for the biological people. So to take it forward, how we'll take the call. So which one is the immunogenic peptide? Now we should take some values, because obviously with the screen itself, sometimes we could see the some spots are really very intense. So this might be the very interesting signal for us. How we could do that? How we could data point of view, how we could take it forward. So we need to establish that. So for that, what would be our call? So Z-score we need to find out. So Z-score we already find out. If now which one can be taken as the outlier? So we have taken a call of the Z-score because some points are really a very outlier. So if you can consider the Z-score greater than 3. Why greater than 3? Because mu plus 3 standard deviation. It is the concept. So after you scaling the data set into Z-score, mu becomes 1 and sigma becomes mu becomes 0 and sigma becomes 1. So if you replace with mu equal to 0 and 3 into 1, it will give you the value of 3. So above the value of 3, this will the signal which we are getting, That could be our interesting immunogenicity, that is making our particular concept of immunogenicity. So this way we have taken, so everything has been done, and data visualization point of view, R is very important. So now you could see we have developed this kind of particular heat maps in R itself. So now after that, after the immunogenic peptides and epitopes, so epitopes concept I already given into screen itself. So you could see the contiguous peptides, simultaneous peptide you could see. So that would be taken as the epitope. If immunogenicity lies for continuous of these peptides, then it would be taken as the immunogenic epitope. That we also reported. Now coming to the next part is discriminatory potential of the peptides, because this is also important for the data analysis. How we take the call. So protein microarrays point of view strategy is different. Peptide microarrays as we are taking with the Z-score technique, now we should very clear about it, which the statistical methods we should take. So we have taken Mann-Whitney test because it is a kind of a non-parametric test. So non-parametric, we are dealing with the Z-score, so we should follow the non-parametric test. So for that Mann-Whitney test came up, and you also know that we usually take up the p-value criteria less than 0.05. And after that, we wanted to find out the significant peptides out of it, as well as significant epitopes. So this way, and this is the algorithm you could see. I already mentioned that data extraction is R, background correction. For R, what I found, so why R? Because of this kind of background correction. Otherwise, you could take it in the Python as well. But for the background correction, because Mann-Whitney is also available in Python. So, but the researcher should take the call which statistical significant test they will provide for this particular analysis because of the parametric or non-parametric test. So, as we have to be dealing with the non-parametric test, so we are taking up the Mann-Whitney test for doing that. Now, after that, this is our new work. So, whatever I told you, that is already we published that under supervision of Professor Sanjeeva Srivastava. Now, this is a new work. So, now last time which I showed you, that is called the severe versus non-severe patient sample analysis. So now what will happen, let's say we are taking some control samples or healthy samples. So that is called a new concept of the cross reactivity. So this time we are working with our team, with the team. So there are huge samples we are dealing right now. We are looking at the IgG profile for that. So now we need to see particularly for cross reactivity part as well as different other studies as well. Now you could see why the images, gridding of the spots is important, because after getting this kind of box spot, this is kind of a discrimination. It is giving us the discrimination that for naive patient point of view and the severe 2 patient point of view, that particularly the non-severe patients, so how they are looking into the box plots. But we need to check it using the raw data point of view, using this particular snaps. So for that, you could say these are nothing but the samples. Every sample's images. When we get any kind of, we are not reporting it because we are in the manuscript level at this point of time. So that's why I mentioned it as peptide X only. So that peptides came significant between this and particular naive and the severe patient. So now we could see it by the itself with the screen itself. So if we are claiming this part is quite means kind of a showing severity, just see this kind of trend with this particular box plot and this particular image as well. So you easily cross verify the data with this particular images as well. So now there are different strategies also there. We could report it in our manuscript, like the mutant panel that was very novel in our part. I'm not, because of time issues, I'm not going to show that. Also immunogenic epitopes panel, all these things we have found in the using data analysis. So this is all about for the peptide microarray work, and if sir will take up. Thank you.
All right, thank you, Arko. And again, by showing the examples of SARS-CoV-2 peptide arrays, Arko have tried to give you the the intricacies of data analysis, and actually a lot goes into really trying to make the sense of the data, right? And that's where our team actually works very hard of presenting data, like how to interpret the data. So you have seen that one example, but again, we don't want to just make it totally biased, that it is only going to be SARS-CoV-2, but actually one could do many other applications as well on these chips, right? So another example which I'm going to show you is about the cancer, which is of course many of you will be interested to study. So most of the time the autoantibody screening of different diseases, various autoimmune disorders, but also sometimes cancer is essentially done to investigate using these kind of arrays. So what you have seen in the slide here is essentially how patient serum can be taken, And from there, one could try to apply the very small aliquot of the serum on the entire high density array, right? And those entire high density array could be then further, taken to look for which of these spots are lighting up uh, for the, binding for the various type of autoantibodies. And you are using either, Psi3, Psi5 or different type of fluorescent tags to visualize the spots. So something like, this kind of workflow we followed for different cancer type, especially brain tumors. And that has really, yielded us some of the early detection markers, which can be utilized for, eventually maybe early screening of the cancer. So this is just one of the platform like protein microarrays, right? But moving beyond the arrays, uh, the next thing which comes is essentially, how to not only do the, binding like this kind of activities, but rather can we also look at, how the interactions are happening and could we measure their kinetics as well, right? So therefore, then there are other platforms which come forward, which not only tell that, okay, there are antigen antibody interaction happens or protein protein binding happens, but how it happens, and therefore they look for the on rate and off rate and the overall KD values. So there are different type of biosensors and platforms which are emerging, and one which we are going to show you now next will be the BLI, biolayer interferometry based platform, which Nirjal Banerjee is going to show you directly from the lab. And he's going to very briefly give you explanation of the concept and then actually from the lab show you the demo of that. So Nirjal, over to you now.
Hello everyone. This is the biointerferometry instrument. This is the biointerferometry instrument. So here we can perform all kinds of interaction-based studies, two molecule interaction-based studies. So its principle is basically on the analyzing the interference pattern of the incident white light that reflects on two surfaces. One is the this tips its own surface. This tip has its own surface, and then there is a chemistry surface which is based on each tip. Either it has streptavidin coating or it has various other coatings. So those two layers and interference on those two layers decides the actual data of this instrument. So here basically we can assess any binding kinetics of ligand and molecule based interactions. So the mainly the dissociation constant determining. So the association rate and the dissociation rate, the ratio of it. Also, it can be used for assessing the steady state affinity and kinetics of any interaction. Again, relative and absolute sample quantification can also be done, provided we have the known standards of those molecules. And finally, epitope binning, which is basically knowing the different epitopes of a similarly binding antigen and antibody. So now moving towards, as you can see in this instrument, so here we keep all the samples of it, and these are the tips. So basically, the instrument takes the tips and dips into several of these wells where we can keep the samples. Again, moving back to the software, how it is. So first of all, we designed this 96 well plate. So again, this is a black 96 well plate so that any other light doesn't affect. So here you can see the loading, basically the one molecule that binds to the tip and acts as the ligand. This is called the loading, and we definitely want to keep some controls. So without any molecule of the loading. So these are the things, and these are the samples of the different concentration, the analytes with which this loaded molecule will interact and give us the interaction ratio or the interaction data. So like this, we can denote all the steps that is necessary, the baseline, the loading, association, dissociation, etc. And also we can denote the sensors and so on and so forth. Let us give an example of how a data after the experiment looks like. So first we have to denote which of them is the reference sensor I, the no molecule bound sensor. Also the reference sample if we have a zero concentration sample, and After that, certain data correction and where we need to align the data, baseline, association steps, etc., and the ratios. Finally, we get to the result. Either we want to see both association dissociation or either of them, and what kind of fitting we want to see it, what kind of ratio they are binding into, and so on and so forth. Finally, we see these kind of, so these are the different concentrations of the analyte with which the ligand is binding. So these kind of different data points we have. So here by the data points, we mean the shifting of wavelength denoted by nanometers. So every incident light, when it interacts with another molecule, the reflection of it changes, and by how much nanometer it shifts, the interference increases, gives the actual data of us. So that's how we can also calculate the KD, which is basically the dissociation constant obtained by K small d by K small a, the dissociation rates and association rate ratio. Also, we can figure out a steady state equilibrium, which is basically generally for the enzyme kinetics. We can find out, which is also denoted by KD only. People might have heard that during the enzyme kinetics. Again, coming back to one example data, one published example data. As you can see here, this is how they have planned the experiment. This is how they have planned the experiment on the left. So first of all, antigen loading. Antigen here is the loading, i.e. the ligand. After that, a washing step is necessary. And then the analyte, which is here the antibody present in the serum samples. So they are trying to see how much antibodies are present against a particular the SARS-CoV-2 antigen. And then finally the detection. So here on the right, you can see that there is a huge significant difference between the seropositive. That means the individual that has been infected before and now are immune to the disease and have the antibodies of those, how much difference their sera has as compared to the seronegative, the people they have not been introduced to the disease, they have in terms of the quantitation of the same antibody present in the human serum. So similarly, the basic thing is any kind of interaction, molecule to molecule interaction can be performed here. After several optimizations, not just only more than two molecules can also be used to find their interactions. Many other plethora of applications can be performed in this beautiful experiment. So it is a modern version of many other similar kind of experiments like SPR. It can also do the likes of what ELISA does. So any kind of interaction, that's the thing. Thank you.
All right. Thank you, Rajir, for explaining the BLI and giving the overview of the technology and the concepts. Next, we are going to talk about one of the high throughput screening platform, SMC Pro. Essentially, while you have already identified some of the key molecules which you are going to measure for various type of clinical applications, then you need high throughput platforms and high throughput screening, and therefore this in fact new tool which we have in our lab can be utilized for high throughput screening. So Surabhi is going to give you an overview of this technology very briefly. Surabhi, over to you now.
Giving you a brief introduction about an instrument that we have, that is the SMC Pro, single molecule counting. So as Professor Sanjeeva mentioned, that now that we have identified some proteins or some molecules that we might want to quantify in actual clinical samples, for example, serum or any other sample. So this particular instrument has very high sensitivity. That is, it can measure these molecules at these proteins at femtogram levels. So the principle is mainly like ELISA. So there is antibody-based detection of the protein that we are looking at, and for example, so like this is a 96 well plate in which the experiment takes place, and there are magnetic beads which have antibody against the particular analyte that you're looking at to pull down those proteins, and rest of the sample is washed off, and then using another antibody, the analyte against the analyte, which is also fluorescently labeled, the secondary antibody, that is used for detection. So after the experiment is performed, like the entire experiment is performed in this plate, the after appropriate washing, the analyte which is to be analyzed in this instrument is taken up, and it is added to this particular plate. As you can see, it has very small wells, so that the even small volumes can be analyzed, and therefore that is how even small levels of the analytes can be measured. So as you can also see, this is there's a glass slide at the back, so it is transparent, and like this plate is inserted in the instrument over here. So this is the software, and this is the plate layout. So we can give the plate layout to the instrument, and it will measure the absorbance in the wells, and then again with a standard curve, it will calculate the concentration of the analyte or the protein that we are looking at. So that's about SMC Pro. It's called single molecule counting. And thank you. Over to you, Professor.
All right. Thank you, Surbi. I think it was very nice, quick overview. So let me now, talk to you about one more technology, and that is SPR, surface plasmon resonance, which is one of the key technologies. Avinash, could you unshare yourself?
Yes, sir, I did that.
Mm-hmm. All right. So, one of the technology which has been in the field from long time, the major biosensor for measuring the molecular interactions is surface plasmon resonance, where on the gold slide we are, having uh, this, on the prism we keep this gold slide when all the analytes have been printed. And now when we want to, flow a small molecule or the protein of interest, they are going to bind to this molecule printed on the chip. And if there is any binding happens, then you are going to see the change in the refractive index of the medium. So this is, what you are seeing in the sensorgram, where initially there is a static baseline, and then it moves to the, binding starts, which is the association rate or the on rate. And then after the binding is completed, and you keep flowing the buffer, then dissociation start, which is known as the KD, K small d, and followed by you can do the regeneration of the chip. So this whole thing is the SPR sensorgram. This is, I think much clearer explanation of the principle here. When we have the prism, the light source, the antibodies are printed and the protein molecules is coming, some of the proteins are binding. And what you see here is the sensorgram of different concentration of this molecule. So this is actually quite widely used technique for the doing the biomolecular interactions, and additional approaches like biolayer interferometry are coming forward to to improve in some ways. For example, here one could go do only few interactions, I think 4 or so in the channels, whereas then now you can look into the plate based entire molecular interaction using the BLI approach, and different principles are involved. So one need to look for complementary approaches. Additionally, one could also try to employ the protein array based density, which is achieved in the high density format on the protein arrays, and then implement that with different type of other label free biosensors. Especially there is a more emphasis coming with the SPR imaging based platforms, which employ the array kind of format and take the pseudo image of the array and look for the binding interactions. And similarly, many other principles of the various type of label free biosensors are coming forward with the carbon nanotubes, planar waveguide arrays, spectral reflectance, imaging biosensor, nano hole arrays, etc. Let us now have a quick demo of the subfield carbon resonance SPR setup in our lab, which is Bioco T200 and shown by Rucha in this video.
Sensor chips, biosensor chips, on which we immobilize one of the interacting partner and we pass the other partner. The partner which we immobilize is called as ligand and the other which we pass is called as analyte. So in Bioco T200, here is the chip compartment where we drop the chip. Okay, you put your chip here, and this is sample compartment. We'll put your sample here, and this is buffer compartment, and this is discard. So you immobilize your one of the interacting partner, and then you will pass the other partner, and you will get the association and the dissociation. So for immobilization, you use two types of strategies. One is capture and other is direct. Either you can directly immobilize one of the interacting partner or you capture. For example, in direct immobilization, you use different types of chemistries like amine coupling, thiol coupling, aldehyde. So for example, in amine coupling, your amine group of your protein is important. So this chip has a carboxy mechanism. You activate that group and this activated carboxy methylated group will bind to the free amine of your protein. And your protein will get directly immobilized. For example, in capture method, you have nickel NTA on your chip, for example. Also, nickel NTA will, so this step protein will get captured on the nickel NTA. So this is how you immobilize your one of the interacting partner. Then you will pass the other interacting partner. Then you will get association. Okay? That rubber will pass. This will give you dissociation. The ratio of this association rate constant and the dissociation rate constant will give you dissociation, will give you equilibrium dissociation constant. That is the strength of your interaction. So after passing of your analyte, if your analyte is not getting completely dissociated, you will need to regenerate. For that, you have to use different regeneration buffers and you have to optimize that. So after regeneration, you will get the sensogram like this. So for example, this is the association, this is the dissociation phase, and here you regenerate. Okay. So you have different types of chips are available. On basis of what experiment you want to perform, you will choose the chip. For example, if you want to perform, you want to immobilize his stack protein, you will use NTA. If you want to immobilize lipid, you will use L1. If you want to immobilize biotinylated nucleic acid, you will use acetyl. This is how, on basis of your experiment, you will use different types of chips. Now, what you get? So, when you pass your analyte, you will get the association. This is how the association looks, and then when buffer will pass, we will get the dissociation. So, this will give you this will give you a small Ka, the association rate constant. This will give you the small Kd, that's it, dissociation rate constant, and its ratio will give you bigger Kd. That is dissociation, that is equilibrium dissociation constant. That means this will give you the strength of interaction. More is the strength, smaller is the Kd value. Okay. So why do we use SPR? Like what extra information SPR is giving? So you can get Kd value from other assays also. Like for example, by agarita, for example, you can get the Kd. Then why SPR? So if you can see here, there are if you can see there are different types of sensograms. Okay, so you can see the shape of the sensogram. This. So this is in this example. This five have the same KD. The KD is same, but this association constant and the dissociation constant is different. For example, if you see here, the association is very. For example, in this green sensogram, you can see the association is very fast, but the dissociation is very slow. Here in blue, the association is optimum, dissociation is slow, but as compared to green, it is still fast. So this information will reveal lot of information. You have a same KD, but different K, small k and small KD. That is, this will give you like different K and KD. So this is the example. What we these are the different sensogram. These are the different interaction study example. For example, in this sensogram, the KD is similar. Okay, you get the KD in nanogram. But if you can see the shape of the sensogram, see even here, association is fast, but dissociation is very slow. Here, the association is optimum, dissociation is slow, but as compared to this, dissociation is faster. So this information will give you a lot of information. For example, when you are designing a drug, in designing a drug, you have to be like very much careful about what is the pattern of association, what is the pattern of the dissociation. This will give you a lot of information. Not only the KD will give you the square. So what is the affinity? How strong is the association? How KD will give you the strength, like how strong is the interaction. But this KA and K off will give you the extra information. So by using SPR, you can study the small K, small K off, and that ratio will give you the strength of the interaction. Okay.
All right. So you got an idea for performing SPR experiment. Of course, if you have watched the videos in the course thoroughly, there has been many sessions of the performing SPR experiments, and I'm sure you will appreciate that in a way, like if you can perform these experiments using complementary approaches, you will have more information, more idea that how you can understand the system better. So just to wrap up, uh, there are different technology which we have been studying in the course, and not only proteomics, but even other technologies, including the transcriptomics, genomics, now metabolomics has come forward. So many of these technology together, which generates the omics data set, can be very helpful to understand a system much better. And that is actually pretty much now being used actively for the translational research. And I think that also brings me to kind of a closer. But a student had asked me once a question about that everything is happening in the proteomics now with the mass spectrometer, where you foresee that technologies like protein microarrays, etc., can still be useful. And I think by the end of this course, since you must have studied the things now much more in detail, I'm sure you will appreciate that every technology has its own benefit. And while mass spectrometers are very strong platform, they are able to provide us high throughput information in a very quick manner. At the same time, um, if you think about the complex biological samples, especially human samples like blood sample, serum, plasma, where we have abundance of some of the proteins like albumin, IgG, and we have the many interesting low molecular weight and the low abundant molecules over there. It is really important to get rid of the high abundant proteins before the mass spec can see your low abundant protein, because the dynamic range of the instruments are very less. Whereas in case of protein microarray, we are actually just simply taking the serum or plasma sample, diluting them, and then putting them on the chip. So we are not modifying anything, we are not changing. And the readout can actually go to this 10 or to the 10 power of the magnitude, which is much broader as compared to 5 or 6 order of magnitude for the mass spec. So again, now you can see for the true biomarker discovery program, when especially for the human samples and clinical sample, you might want to consider alternate platforms. Not only all the time mass spec can be only way of moving forward. So therefore, this course has probably given you a balanced perspective of using different technologies, different platforms, in which way you can try to get the best out of these technologies and utilize that for your actual application, biological application, which you are interested in. We wish you good luck for completing this course and the examination, which will be happening soon. And if in any way we can help you provide further opportunities or any kind of further training or interaction, it will depend on your performance in the course. And if you do well, we'll be very happy to further interact with you and try to see if in which way we can give you further training or internship opportunities at IIT Bombay. I hope you have enjoyed the MOOC course. We have really tried to make it as live and as interactive as possible. But of course, given that large courses, many students interact and many are not able to interact as well. But this is actually a good opportunity for you to take the next step forward and try to see if you'd like to learn further and build your career in this direction. So that's pretty much from our side. Let me thank all the TAs, especially Avinash and Surbi, Arko, Nirjar, Argyo Banerjee, Shashwati. All of them have worked very hard, not only for today's live session, but also throughout the course. And the entire team has worked together to try to bring the best in short time possible. I wish all of you good luck, and we'll close from here now. Okay. Thank you, everybody, and bye.